His name is Dr. Rudy Tanzi, and he is a professor of neurology and holder of the Joseph P. and Rose F. Kennedy Endowed Chair in Neurology and Mental Retardation at Harvard University. Dr. Tanzi is the Joseph P. Professor at Harvard and Director of Genetics and Aging Research Unit at Massachusetts General Hospital. Dr. Tanzi has been investigating, just lost it.
I have it on paper here.
Okay, great, that would be better. The old-fashioned way.
Thank you. You're welcome.
Okay. Dr. Tanzi discovered the, let's go back. Dr. Tanzi has been investigating the genetics of neurological disease since the 1980s, when he practiced in the first study to use genetic markers to find a disease gene. Dr. Tanzi co-discovered the 3 familial early-onset Alzheimer's disease genes and several other neurological disease genes, including that responsible for Wilson's disease. As a leader of the Cure Alzheimer's Fund Alzheimer's Genome Project, Dr. Tanzi has carried out multiple genome-wide association study of thousands of Alzheimer's families, leading to the identification of novel AD candidate genes, including CD33 and ADM10. His research on the role of zinc and copper in AD has led currently to ongoing clinical trials at Prana Biotechnology. Dr. Tanzi serves on dozens of editorial and scientific advisory boards and as a chair on the Cure of Alzheimer's Fund Research Consortium. He has received numerous awards, including the 2 highest awards for Alzheimer's disease research, the Metropolitan Life Award and the Potamkin Prize. So thank you.
Thank you so much, Lexi. And without further ado, well, here's Rudy Tanzi.
Well, thanks to Glenda and the Aspen Brain Lab for inviting me for this great event today, but I'm not as grateful for having to follow Mick Eberling. It's like, what? So I'm just going to kind of pack it in and do my best. And thanks very much to Ms. Potamkin for this award. That, you know, the Potamkin Award is kind of like the Nobel for Alzheimer's. And one thing prizes do is it drives people to work very hard. And people work very hard in their research, not just for helping patients for this terrible disease, but also to get awards like that. So they really serve their purpose. I think this mic's a little loud. It might be feeding back. I'm not sure. No, it's okay? Okay, it's just over here. So anyway, I'm going to talk to you today about Alzheimer's disease, which is really such a terrible, terrible disorder, because what it does is it robs you and your loved ones of yourself. So what I think I learned most about not just the science and biology of Alzheimer's disease, but in combination with writing Superbrain with Deepak Chopra, was what does Alzheimer's disease teach us about who the self is? So in essence, all our life, from infancy, from the womb, we're building a neural network, connections of synapses. Every experience we have, we're layering upon what we've already learned, and we're constantly building from a foundation, a neural network of 100 billion neurons, up to a quadrillion connections between these neurons or nerve cells called synapses. And what Alzheimer's does is it takes this rich tapestry that describes who you are to your loved ones, to yourself, and just unravels it slowly. And as it unravels that tapestry, just like when you really do that, as you go further into disease, it unravels it faster and faster. So at first, you realize your own tapestry of yourself is being unraveled. Terrible, terrible thing. And then later, you don't know, and now your loved ones and your family, your friends are just seeing you have no idea of who you are. Self-awareness, self is robbed from you. There's no worse fate. And the scary thing is that this disease is becoming more and more prevalent because we live longer. All of modern medicine has us living longer. The average lifespan is 80 years old. At 85, 50% of people have Alzheimer's. It's just crazy. So unfortunately, we need a lot more funding to fight this disease. I think we're a great example of having a lot of knowledge but being a budget-constrained disease. And I hate to say it, but to some extent, it's because it affects old people. You know, and in Congress, you know, diseases that affect younger people, they pull the heartstrings more. And so there's a real problem with getting enough funding. But what is Alzheimer's disease? Well, Alzheimer's is the most common form of dementia in the elderly. Age is the #1 risk factor. 1 in 5 people over 65 today will get this disease, over 50% over 85. I started studying genetics 30 years ago at Harvard. I'm still there, still studying genetics and neuroscience. And it got my attention that family history was the second greatest risk factor. And the idea was if we could find the genes that cause this disease, we'd have an idea of how to fight it. We need targets for drug discovery and for therapies. Just some facts. Females get it more than males, even when you account for lifespan. Any injury to the brain, bangs to the head, a small stroke, perhaps due to obesity or high cholesterol, diabetes, any insults to the head can help trigger this pathology. And then once you get Alzheimer's pathology, we now know that it starts to spread through the brain like a fire. So just small areas can spread. Loneliness, social isolation is a risk factor. Depression, emotional trauma, losing a loved one, going through a bad divorce. All of these things can cause damage in the brain. With emotional trauma, cortisol can form. And there are over 5 million, already 5 million victims in the US. 13 million family members are impacted. $200 billion a year. $200 billion a year is spent on caring for Alzheimer's patients. And right now, the federal government spends under $500 million on research. And the problem with that, amongst everything else, is that if you take 74 million baby boomers like myself heading toward risk age, and you calculate what % of Medicare and Medicaid does Alzheimer's consume right now, and you do the math, somewhere between next year and 2020, Alzheimer's disease has the capability of single-handedly crushing Medicare and Medicaid. And then you look at a graph like this. Up top, you're seeing mortality rates, AIDS down, pneumonia down, stroke down, heart disease down, diabetes down, cancer down, Alzheimer's way up. Then look at the funding. It's great, cancer, heart disease, AIDS gets a lot of funding, but Alzheimer's is the red-haired stepchild. It's just not getting the government funding we need. I'm very lucky because I work with a foundation called Cure Alzheimer's Fund, and every dollar they get in donations is $1 out. The founders of the foundation pay all the overhead, so dollar in, dollar out. They've been very generous in helping support, together with the NIMH, the Alzheimer's Genome Project, which I direct. And the idea is to use genes to get to the cause of this disease. Currently, the drugs we have, like Aricept, Exelon, Reminol, Namenda, they're better than nothing. They treat the symptoms temporarily, in a minor way, but they don't treat the disease. The disease still ravages the brain while you take these drugs. So they might just help get a little bit more cognition out of a patient. And meanwhile, what can you do to reduce your risk? And these are the things that have been shown through various studies along the way. Physical exercise, healthy diet, Mediterranean diet especially, staying socially engaged, not holding up in the house, learning new things. Right now, you're being protected from Alzheimer's disease. So when you make new synapses, you actually are strengthening synapses you already have and strengthening your neural networks. And in the end, if you ask what correlates most with degree of dementia and Alzheimer's disease, it's loss of synapses. So I tell people when you're planning for your retirement, you save money in the bank and you save synapses in your head. And the way you save synapses is you just learn new things and make new synapses. So you need to think about not only monetary reserves, but cognitive reserves, synaptic reserves as you get older. So you're doing the right thing by just being here. Meditation and yoga, de-stressing, you'll hear maybe more about that if you're attending the session with me and Deepak Chopra today, and sleep. We now know that during sleep, this is where you get a lot of your long-term memory consolidation. You're learning things all day as short-term memories, and a lot of that consolidation to long-term memory happens in this deepest part of sleep called slow-wave sleep. We're also learning it during this part of sleep. This is when the brain starts to clear itself of a lot of gunk and protein garbage that builds up in the brain. I call this mental floss. And if you don't sleep enough, it's kind of like not flossing your teeth, and eventually you're going to get into trouble. Except you're not going to just be fixed with a simple trip to the periodontist. Now, what's a senile moment versus Alzheimer's disease? So here we have, I know I came into this room for a reason. Dinosaurs are saying, oh crap, was that today? Is Noah's Ark? And this is real. This signs are real. Senior Center, don't forget. Senior Center, remember to turn. Senior Center, wake up. Anyway, it goes on and on. But so what is this? So how many times do how many, be honest, have dialed a number on the phone, and then the person's about to answer the phone, you're like, oh my God, who did I just call? Yeah. So what this is in all these situations, when you get into the kitchen and your cat follows and you look at each other and have no idea why you're there, you're doing these things, all of these things, senior moments, it's not that you forgot what you were doing, you never learned it. You didn't register the information. You see, as you get older, we get, bandwidth is higher, we have to do more things, we get distracted. We don't take time to register what we're doing. That's a senior moment. That's very different, and that's fixable. Just focus, pay attention. Be present in the moment. Be like a 6-year-old kid. Wow moments every minute rather than so what moments. So that's fixable. Alzheimer's disease is different. The brain is under attack. So what's happening here is that you start with pathology toward the middle of the brain, and then it spreads and spreads and spreads like a fire. And as it goes, you basically lose the ability to learn. Alzheimer's disease is as much a learning disease as it is a memory disease. And there's specific pathology that causes this. And for many decades, we just could look at that pathology, but we had no idea where it came from. It's like the 5 men in the room blind men in the room feeling the elephant, trying to figure out the disease. So what really happens, and this is the first thing that happens in Alzheimer's, is that an area of the brain, don't worry about the slide, I'll just show you. So all of your sensory information comes into your brain, everything, all your senses are bringing it in. And that information has to be stored as short-term memory in the middle of the brain, in the hippocampus, okay? And there's basically a pathway, a cable, that sends that information to the hippocampus, so that as you're listening to me right now, you can remember the slide I showed, 30 seconds ago. In Alzheimer's disease, that cable that's bringing that sensory information in of everything going on right now, so it can be stored as short-term memory, gets slowly severed by Alzheimer's pathology. So at the end, in the extreme end of the disease, the patient's in a room sensing everything going on in the moment, but that information is just kind of feathering away. It doesn't get stored. And so they're basically living in the moment with no chance to know what happened a second ago. And there are some who say, well, we spend our lives trying to learn how to be present in the moment. Well, the difference is there's no sense of self. Right? You're living in the moment, but there's no context. There's no backdrop of who you already are to relate that information to. And it's actually terrifying. I don't know how many of you seen an Alzheimer's patient in the late stages, and there's literally terror in their eyes. Because can you imagine everything happening and you can't relate it into any context at any given moment? It's just devastatingly scary. And that's what happens in this disease. Now, it's really shown well when, in this first interview, when Alzheimer's first described this pathology in 1906 in Bavaria, in an amphitheater. Carl Jung was in the audience. He was trying to argue that physical lesions in the brain are causing this woman to have dementia. And believe me, people didn't buy it. Mental illness, physical lesions, how can that be? But he described this conversation with her, and she was sitting on her bed, helpless. This is from his journal. And he said to her, "What's your name?" And she said, "August." Good. "Last name?" She said, "August" again. "What is your husband's name?" She said, "August, I think." So she's having trouble. "How long have you been here?" She said, 3 weeks. She had actually been there for 2 days. And then he said she broke down, and she just kept saying to him, "Doctor, I've lost myself. I have lost myself." And that's what this disease is. You lose yourself, and others who have known you your whole life lose that person who was once there, still present in their body. So, what this teaches us is that we have to treat, basically that this disease is a loss of self, and how are we going to stop it? And like I said earlier, my approach, and others have been, find out the genes that cause this disease so that we have actual physical targets that matter, and then use those genes to predict who has the disease early on with proper counseling and protection. Keep track if you're at high risk with a family history by imaging for that pathology in the brain and other biomarkers. And if you're in trouble, stop that disease from hitting before it actually causes problems in the brain. And it turns out that the pathology in Alzheimer's disease begins 10 to 15 years before there are any symptoms at the level of cognition or dementia. So we have a chance to do this. So I'm going to tell you now the hope we have for the future generations of knowing who's at risk and stopping this disease, nipping it in the bud before it hits as a way to eradicate Alzheimer's. I'll also tell you of what we have to do for patients who have this disease right now, because this is obviously not good for them. So let me talk a little bit about this. So it gets to genetics, and this is just a timeline of genetics from the Smithsonian, starting with Mendel and his peas, and going on to the discovery of DNA. And this one here that says 1983, I was really lucky to work with Jim Gasella back in the early 80s. We were literally kids in our 20s, and we were the first ones to find genetic markers in the brain, in the genome, and use them to find a disease gene called Huntington's disease. So that was the first time a disease gene was found using genetics. And it galvanized me to want to do something else, different than my mentor, so I went into Alzheimer's disease. And then later on, the Human Genome Project led to the ability to do what we call the Alzheimer's Genome Project. So if we just go through the history of this disease very briefly, for 80 years, from when Alzheimer's described that pathology, these so-called plaques, these big bundles of what's called amyloid, like boulders that sit outside of nerve cells, the nerve cells then respond to this amyloid and form inside these tangles that choke the inside of the nerve cell and eventually kill it. And then the brain responds to all this cell death and all this pathology eventually with inflammation. The brain's own immune system, which is quite primitive, it's not like antibodies and T cells and B cells. The brain's immune system is at the level of a horseshoe crab or an oyster. Okay, if it sees cells dying, it says there's an infection, it starts shooting out free radicals, oxygen bullets to kill the invader. Well, if there's no invader, what do you kill? Collateral damage, you kill nerve cells. So inflammation is doing that after the pathology accumulates for 15 years. So we had no idea how this all occurred, and then in 1986, I and others discovered this gene I called amyloid precursor protein. It makes the amyloid. 1995, again, others and I found these two genes called the presenilins, which help make the amyloid. And then a group at Duke University found a gene called ApoE, which helps get the amyloid out of the brain. So now we had 4 genes that make 4 proteins that can carry mutations that all taught us that amyloid is starting this disease in these early onset cases. And in this case, these are people with these first 3 genes who are getting disease under 60, under 50 years old. Mutations that guarantee the disease, which are quite rare, but we learn the most from them. So what do we do with this? We take genes. We're not going to go in there and necessarily try to change the genes right away, but that may be in the future with new advances in genomic editing. But you come up with drug targets based on what you learn from the mutations in those genes, and then devise new therapies. So an example is heart disease, right? It started with genetic studies of families with early onset heart disease that suggested, based on what they found, that cholesterol was the issue. And so to manage risk for heart disease, diet, exercise, weight control, and if necessary, people take drugs like Lipitor or Crestor, statins, to lower their cholesterol. The equivalent in Alzheimer's disease is this little protein that George Glenner in 1984 named A-beta, and then I and others found the gene that makes the A-beta. A-beta is this little protein that's made by nerve cells that spit out, and normally it's made in the brain, but if you make too much of it, or if it accumulates too much, it forms those plaques. It also makes these little clumps. So as the nerve cells are trying to communicate, the little clumps of amyloid get in there and actually short circuit communication between nerve cells. In addition, it's causing the connections of nerve cells to break down, and in response, the nerve cells start to form these tangles, and then nerve cells die, and then you get inflammation. So this is what we learned in a nutshell from these very first genes. Now what happened was, how many of you heard or read of the many failed clinical trials in Alzheimer's disease? Raise your hand. Right, so here's what was going on. Now some of those drugs were just bad. Pharma companies pushing drugs for no apparent logic. But some of those drugs were pretty good. And what was happening is, all of them, the middle is a sink overflowing. That's your brain overflowing with amyloid, right? So if you have a situation of an overflowing sink, you try to turn off the spigot, or you try to unclog the drain. So there were drugs that were developed that tried to turn off the amyloid production. Other drugs are trying to clear it out of the brain. I'm working on a couple of these, like these so-called gamma secretase modulators that are working quite well. This other one called PBT2 to clear it out. It's a work in progress. We're in clinical trials. But the original trials failed. Why? This is the reason why. Amyloid starts 15 years before the disease actually strikes. So what happens here is that if you follow that red line up for amyloid, when it starts to plateau and get flat, that's when you have dementia. So what we were doing was we were treating patients way down on the right who had dementia to stop their amyloid when the amyloid already did its job. It was too little, too late. It's kind of like having a patient have a heart attack, has congestive heart failure, and they come to see the doctor, cardiologist, and he says, that's okay, just take a Lipitor, right? You had to do that 10 years before. So now we know that you have to treat amyloid 15 years before symptoms, and now we can image for that amyloid and start thinking about how to do that. The tangles start a little later, but for a patient who has a disease right now, what's really pushing them off that cliff is inflammation, the brain's adverse response to that pathology, seeing these dead nerve cells freaking out, the immune system in the brain freaking out, saying, all these dead nerve cells, we must be under attack from a bacteria, a virus, a fungus. This is the same thing the brain's immune system did 25,000 years ago when we were running around the jungle. And now the brain's trying to kill something that's not there, and it's just killing more nerve cells. That's the disease in a nutshell. So how do we fix this? Well, we need more genes. So in this Alzheimer's Genome Project, now with the first whole genome sequence costs, what, $2 billion? Now we can do a whole genome sequence for $1,000. So quite a nice discount. So we've been carrying out whole genome sequencing of members of Alzheimer's families. We've been looking at brains full of plaques and tangles, but they didn't get the disease. So we're saying, what protected them? And we're finding that a lot of these people who have the plaques and tangles but no disease have genes that protected them against the inflammation. We're learning from that. We generated 0.5 petabyte of data. It's a data nightmare. We spend, I don't know how much of our money now, we spend more money on CPU time, buying CPU from Amazon, than we do on lab reagents, because we have to crunch this data is so CPU intensive, and Harvard's research computing facility, we can't just hog all their CPU, we have to buy it. So this is what we budget for now. It's a big data issue. But the idea is if we can find all those different genes that cause this disease and all these different variants and learn how they do it, we can do something like this. There's the amyloid. We found those genes are involved there. There are the tangles down on the left. Those genes are involved there. Inflammation is these genes. Now we actually have physical targets, genes and proteins. We can learn from the mutations in those genes to say, how do we treat the disease? How do we prevent it? And we're making great progress. And this is basically where we're at. We have a paper in press in the journal Nature as of this past, already? Now, wait a minute. I only had 24 minutes on here when I started, so, but all right, fine, I'll be fast. Okay, so we have basically come up with what's called Alzheimer's in a dish. This is a real breakthrough. In 6 weeks, using a gel that mimics the brain, we can get plaques, tangles, full-blown Alzheimer's pathology following the correct course, as it does over 80 years in a brain, we do in 6 weeks in a dish. And now this paper is now in press, so now we can just test all the drugs we want quickly and ask which ones stop the plaques, which ones let the plaques form but stop the tangles downstream of that. So it opens up a whole new era. And in the past, we had to use animal models that didn't even recapitulate the whole pathology, and it took a year or two. 6 weeks per drug, and we can do it in a 96-well dish and have a little mini brain in each well of a 96-well dish, test many drugs at a time. Thank you. Thank you. So here's an example. The plaques are forming in 6 weeks. We add this drug that I'm working on, the plaques don't form. Example of tangles. We found out that now we found the target to stop the tangles. We can have all the amyloid you want. The nerve cells don't make tangles. You intersect at that moment. These drugs do that. And so basically, we found also for the immune system how to target these two proteins so these little cells that are normally helping the brain in green don't convert into these soldiers that attack the brain in red. We figured out the switch. CD33 is the switch. Now we're trying to hit that switch so that you can have all this pathology, plaques and tangles, but if your brain's immune system doesn't turn against your brain, you save the patient. So now we're figuring out how to turn that protein off. Another target there. So we're getting there. This is just showing that Now we can think up top, that's how you prevent the disease early on. Stop the amyloid, get it out of the brain, stop the tangles, stop their spreading. If a patient has a disease now, you go to the bottom, stop the inflammation, and all these genes are teaching us how to do that, and now we have the Alzheimer's in a dish to test these drugs quickly. So I'm very, as you can tell, excited and optimistic about the future. In the end, we're going to need a cocktail to hit all these different pathologies, but according to the University of Bordeaux, we just need a glass of wine. So we do that. And that leads to this cartoon, which is, where this neurologist says, red wine makes them live longer, but they get to be a real pain. And this mouse is saying, it has a timid nose with the usual notes of oak and vanilla. And this mouse says, I can't believe you're drinking Merlot.